Give everybody a couple minutes to get in. I didn't post the link until the last second, so some people may not have seen it yet. And so give me 1 minute. I'm gonna go let my dog out real fast. He's gonna bark the whole way through. Give me just a second. All right. Let me make sure I got everybody. All right, guys. We're gonna get started on the next chapter, chapter 4. I also have that little bit of chapter 3 embedded in here when we go over the staining techniques. That's all you'll need to know from that chapter. Let's see how far we get today. I know we have your test scheduled for Sunday, but I may put it off until Wednesday. So we'll have one more class period to kind of finish up if I don't get through with everything today. And I doubt that I will. So we'll play that by ear and kind of decide at the end if we get through or not. So anyway, what we're going to look at now are cells. Some of you had questions about the extra credit. You can find that underneath chapter 4 handouts, the cell model. It goes along with this chapter. So if you choose to do that, it's going to be found there. And the timeline is underneath chapter 1 handouts, right, in case you can't find those. Anyway, we're going to start out with looking at cells. There's two major types of cells that we have. We have prokaryotic cells, and that comes from the word pre-nucleus in Greek, and eukaryote from the Greek words for true nucleus. So prokaryotes, no nucleus. Eukaryotes have a nucleus, and prokaryotes are going to be your archaea and your bacteria, whereas eukaryotes are everything else, including us. So full comparison, prokaryotic, the chromosome is different. We'll get into this in a little bit. We'll talk more about the chromosome and how it's shaped, and then we'll get into the genetics chapter, and I'll go over it in real detail. But there's no histones that hold the DNA together. Prokaryotes have no organelles. And bacteria are going to have something called peptidoglycan in their cell wall. And that's something we're going to look at closely. And we'll do the Gram stain. I'll go over how that works and tell you why it matters, which type of cell wall it has. The archaea are going to have what's called pseudomerian cell wall. So they don't have the peptidoglycan. Again, these are two different types of organisms completely. They used to be lumped together because they are prokaryotic, but the archaea and bacteria branched a long time ago. We now know that they are two distinct domains, so we're not putting those together anymore, even though they look alike. Um, Eukaryotic cells, these are going to have paired chromosomes and a nuclear membrane. We have histones, we have organelles, polysaccharide cell walls when present, like in plants. You have chitin cell walls like in fungi. And these are going to divide by mitosis instead of binary fission. These are very tiny cells, and most bacteria that you're going to be viewing are around 0.2 to 2 micrometers in diameter and about 2 to 8 micrometers in length. Again, you'll need to put these under oil immersion to be able to even see them. Again, some of these are like BMAG is fairly large. So you'll look at it under the microscope, you'll be able to see it in a lot of detail. But some of the others are very, very tiny. But this is what you're going to start seeing in lab when you start looking at these guys through the microscope. You're probably going to start with a simple stain. I don't know if y'all are there yet, have done that one. But you'll start out with simple stain, then you'll get into the gram stain, acid fast stain, endospore stain, capsule stain, and you'll do all of these techniques. And when you stain these bacteria, it makes them stand out so you can see them. If you were to put them under the microscope without staining them, they're clear, you wouldn't be able to see anything, right? So this stain is going to lock into that cell wall, and that's going to make the bacteria visible under the microscope. Now, there's several shapes that we're going to look at, and I've also put one on Blackboard. There's a handout. You might want to use it to study from. The three major shapes are going to be coccus, which is spherical, rod shape, which is bacillus, and then we have some oddballs that kind of don't really fit in. They just kind of have their own unique shape. So we have a star-shaped bacteria, rectangular bacteria, and then we have the spiral bacteria. So the main ones you're going to be looking at in lab are going to be the bacillus, the coccus, and the spiral. And spiral, you may even classify them down even further into Vibrio, which is basically like a bent rod, Spirillum, which is like a corkscrew, and then the spirochete, which is a long, thin spiral type shape. Now, when we look at arrangement of bacteria, you're going to notice when you look under the microscope that some of your bacteria will be found in chains. That is strep. You may find diplo, where we have two cells that are fused together. Tetrad is 4 cells fused together, and Sarcina is where you have 8 cells fused together, almost like a cube shape. And then staph is like a grape cluster. These two would be the ones that you see the most of, strep and staph. And you'll have to, in your lab manual, they'll ask you for the morphology and arrangement. Morphology in this case would be coccus. The arrangement would be staph or strep. All right. So you'll be asked that quite a bit. So make sure that you know the difference between morphology, which is the shape, and then the arrangement, which is how the cells are put together. All right. Now with bacillus, same thing. We can have a single. We can have coccobacillus, which is like a cross between a bacillus and a coccus. We can have diplobacilla, and you can have streptobacilla. All right. So again, these are some other arrangements that you might see in lab. Now, most of your bacteria are going to be what's called monomorphic, and monomorphic just means they're the same shape all the time. It gets confusing when some of these are pleomorphic, which means they can take on many different shapes. So you can't just look at the shape and say, okay, I know what that is, because they can look totally different. So again, the same species of bacteria may appear as a coccus in one stage, and then it may appear bacillus or elongated or vibrio in another. Especially sometimes as these things age out, if you get a culture and it's not brand new, they start taking on some different shapes than they normally would look like. So again, you have to be really careful. But lucky for you, most of the bacteria we use in lab are monomorphic. So when you see them, they're going to look the same every time. Right? In fact, almost all bacteria that you use in the lab will do that. Might be some that will take on a different shape if they get old or if the cultures get old. Right. So if we look at bacillus, you can see the shape here is that elongated rod. This is a Gram-Stained Bacillus anthracis, which causes anthrax. You can also see these little green endospores inside of these cells. So these cells are old. They're getting ready to basically turn into an endospore and go dormant. They've ran out of food. They've ran out of places to grow. And once they get old, they start producing these. And you'll do a stain where you'll be looking for these little green endospores. They can be inside the cell or outside the cell, depending on the age of the culture. Now, if we look at a typical bacteria cell, and if you're going to make your model, you can kind of model it on this if you want to, or you can look up any bacteria cell online, get the shape of it. But this is kind of a generalized cell here. But if you want to do something like E. coli or Salmonella, you can look that up, and they have distinct shapes. They may have flagella and cilia. They may have all kinds of different features that you might see on a specific bacteria cell. So if you want to do, again, E. coli or something like that, look up a picture of that exact bacteria. But if you just want to do a generalized cell, you can use this as a model. So if we look at our bacteria cell, we have our cytoplasm in here. Remember, these do not have organelles. And then their DNA is in a circle. Now, you can't really tell here because this is all jumbled up, but if you started at one end and went all the way around, you'd end up back at the same place, right? And then you can see this little circular plasmid. These are going to be important for antibiotic resistance, various things like that that they carry on this plasmid. It's like a little extra piece of DNA that they can turn on and turn off. And they may turn on and create an enzyme that helps them break down a new source of food if their old source runs out. So let's say they use up all the glucose and they want to start fermenting lactose. They may switch on a gene that makes lactase, which is the enzyme that helps break down lactose. And they can do that and turn that on, turn it off when they need it. You don't want to make an enzyme unless you need it because it's a waste of energy. So they turn that gene off when they're not trying to consume that lactose in this case. But when they run out of food, they need to switch it on so they can start utilizing a new food source. Right? So in here, you can see the 70S ribosome. We have an 80S ribosome in eukaryotic cells. So if we have an antibiotic, and some antibiotics work this way, that will attack a 70S ribosome, but leave the 80S ribosome of eukaryotic cells alone, then we can use that against the bacteria to kill it, and it won't harm our cells because we have the 80S ribosome, and that particular antibiotic doesn't react with that. Right? Notice we have a cell wall around the cell, and we also have that cell membrane on the inside of that. Now, the glycocalyx is a sugar coat. It's viscous and gelatinous, and it's made of a polysaccharide or a polypeptide, which is basically a small protein. And there's two types. We can have a capsule, which you're going to look at, and you're going to stain for a capsule in lab, and it'll look similar to what you see up here. It'll have this halo around the cells, and that shows you that there's a capsule around that cell. Or they can have a slime layer, which is kind of unorganized and loose. But this can contribute to virulence. And if it has a capsule, it's an extra protection against antibiotics. It prevents phagocytosis from the immune cells. These can help form biofilms that we talked about before. So again, this can lead to virulence or resistance to death. It makes it harder to get rid of. So if you get infected with this, it makes it harder for the antibiotics to work and that kind of thing. Right, flagella is here, and flagella in bacteria are kind of unusual. They are on almost like a little motor, and they spin around this way, right? So it's going to rotate around, and it's not going to go back and forth like you would think of with like a sperm cell. So when they move like this, and I'll show you some video of them under the microscope so you can watch how they move. They do what's called a run and tumble, where they'll run and then they'll fall over themselves, and they'll run and they'll fall over themselves. And you'll see that again in a video here in a little bit. But this is made of flagellum, and this can even act as a antigen so that the immune system can pick up on it. You can actually, your immune cells can recognize the bacteria flagella and then realize that it's not one of our cells and then start to try to attack it. So as you can see, this flagella is round, and you can see here on the gram-positive bacteria, we only have one of these basal bodies, but on the gram-negative, we have two of these. And this is going to allow that, again, that rotation of this. So the filament is the outermost region that you see. That's the part that kind of whips around. And then you have the hook that attaches to the filament. And then you have the basal body, which is a rod and pairs of rings that anchor the flagella to the cell wall and membrane, and then provide a rotation point for that flagella to move. So this is going to show you a flagella in action. This is a simulation, but you'll be able to see some of the movement that these bacteria make.
Although flagella may appear to undulate back and forth like an eel swimming through water, they actually rotate 360 degrees, clockwise or counterclockwise. The basal body spins, much like the drive shaft of an electric motor, producing this motion. When the flagellum rotates counterclockwise, the bacteria runs in a single direction. When the flagellum rotates clockwise, the bacterium tumbles and changes direction. Movement toward or away from a stimulus is called taxis. The stimulus can be light in the case of phototaxis or a chemical in the case of chemotaxis. In either case, the bacterium contains receptors which send signals to the flagella. In the case of positive taxis or motion toward the stimulus, tumbles become less frequent when the cell moves toward the stimulus and more frequent when the cell moves away from the stimulus.
Right, so this gives you an idea of how they move. Now, when we look at flagella, you'll need to know this for your test. Let me check something real fast. I think it might be on that handout that I gave you. Let me look under chapter 4 here and see. I know the arrangement is there, but your flagella is on that sheet that's in chapter 4. So make sure you know what these are. Atrichus is going to lack flagella. Paratrichus is going to be distributed all over the cell. Then we have polar flagella, where it's at one or more ends of the cell. So monotrichus, single flagella at one pole. Lophotrichus is a tuft of flagella. And then amphitrichus is flagella at both poles of the cell, like you see here. All right. So make sure you know these. You're not going to probably do the flagella stain in lab because it doesn't work very well. And we've kind of omitted it over the years, but you will still need to know these arrangements though that you see here. All right, so atrichus, paratrichus, monotrichus, lophotrichus, amphitrichus. And again, it's on that sheet, that handout that's in chapter 4. So you can just use it to study from, and it'll remind you to know those. Right, so we mentioned taxis a minute ago in the video, but chemotaxis is movement towards or away from a chemical. And we call that positive chemotaxis if it moves towards something. If it moves away from something, we call that negative chemotaxis. And then phototaxis is movement towards or away from light. Yes, that stuff fair game too. So yes, and you can watch it again. If you replay the lecture, you can go back and watch that again if you need to. All right, so again, flagella are going to rotate to run or tumble. And these can act again as antigens, and that can help us to even pick out serobars or variations within species. So E. coli, again, the good stuff is in our gut right now. We have E. coli doing what E. coli does. It helps break down our food. But if it's O157H7, then that one causes a toxin to form, and then that can make you sick, very sick. So again, there's different serobars or different variations within the species. So again, like E. coli, there's more than one type, and some of these, again, like this one, will produce a toxin, again, that can make you very ill. So the run and tumble, again, they're going to just move, 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 and then they're going to change direction, and that's where that tumbling comes in. So again, the run and tumble is kind of the classic way that bacteria move around the media. So this is going to show you bacteria, an actual lot of bacteria, so you'll be able to see the run and tumble that occurs with them. All right, so it just gives you an idea of what bacteria really look like under the microscope if they are moving. Now, yours in the lab are going to be dead, so they're not going to be moving, right? Anytime you stain these bacteria, you basically are killing them. You know, when you heat fix and all of that stuff, you're not going to see them move. So you'd have to have a live culture and have that under the microscope to see it. I don't know if your lab instructor will show you live bacteria like that. Sometimes they do, sometimes they don't, depending on who you have. Um, so again, that's the run and tumble that we see with bacteria. Another one, and I know last semester Crystal brought some of this in, but Proteus does something called swarming, and basically they will make these concentric rings on the media. It's kind of cool to see when you look at it in person. They make this swarming motion where they move outward, and they usually stop and form a ring around it as they grow. So as this colony gets bigger and bigger, they just move out, and we call that swarming. And these have over 1,000 Paratrichus flagella on each cell, so they are really good at moving around because they have so many flagella. Now, endoflagella are found in spirochetes. These are those spiral bacteria, and I'm going to show you some of these moving here in a second. But they're anchored at one end of the cell, and then the rotation causes the cell to move like a corkscrew in it. So it's a jittery type move where it's kind of a real quick little burst, right? I think I have a video of these moving in a second. But this axial filament, again, is an internal flagella, and it basically contracts the cell down and it makes it move in that corkscrew fashion. Let's see if we can see that here. Yeah, you'll see the spiral bacteria. This looks like some algae or something that's in here, but pay attention to the little spiral bacteria. That's the one I want you to watch. Save a little time. I'll cut that short. It just shows you the same thing over and over again. So. Right. Now, fimbriae and pili are hair-like projections. The fimbriae are hair-like appendages that allow them to attach to a surface. And this is composed of something called pilin, arranged helically around a central core. And then the pili are longer than fimbriae, and these can be involved in motility, with gliding and twitching motility, but they're also going to be useful in conjugation. And that's where bacteria can actually switch DNA with one another. So one cell can give some of its DNA to one bacteria cell, and then that one can give some back to the other, and it helps to make variety. So again, we're going to see this because remember, bacteria don't go through sexual reproduction, so there's no mixing of genes. This is the only way in which they can really get some new genes into the gene pool is by going through conjugation with another bacteria and basically swapping little pieces of DNA. You're going to see this too with transduction. Bacteria can actually even grab dead bacteria DNA and make it part of their own and actually change their genetic makeup. So let's say one cell is going to produce a toxin. It has a gene to produce a toxin, and this other cell doesn't. And the cell that produces the toxin, let's say it dies, the living cell that doesn't produce a toxin can actually go grab the DNA from the dead cell, put it in its own, and then start producing the toxin, right? They can do this with resistance genes, so it makes them resistant to certain antibiotics. They can pick that up. And we'll get more into that when we talk about bacterial genetics in another chapter. But it's kind of neat that they can do that. They can swap around DNA. We can't do that. I can't go and pull some DNA out of my chest and give it to you, but these bacteria can. So it makes them unusual. Now, the cell wall is made of peptidoglycan in bacteria, and we're going to look at where that peptidoglycan is located. Do we have a thick layer that's on the outside, or do we have a thin layer on the inside and another layer on the outside? But this is going to contribute to pathogenicity, which is the ability to cause disease. And this is the site of some antibiotic activity. Penicillin works by breaking this peptidoglycan down in the cell wall, and that makes the cell weak, and then the cell dies. So again, we need to look and see where this peptidoglycan is located, and we do that through what we call a gram stain. Right? You're going to do this in lab, and you'll do this so many times that you'll dream about it. Because every lab from then on, after you do that, when you do your unknown and all that, you're going to be doing gram stains over and over and over again. So you'll get the technique down pretty quickly. All right, so this is just a generalized bacteria cell again. So peptidoglycan is a polymer of repeating rows of NAG and NAM, all right? Acetylglucosamine and then N-acetyluramic acid are the two, but we call them NAG and NAM for short. And these are linked by polypeptides, and penicillin interferes with that final linking of the peptidoglycan rows, and that weakens the cell wall. And then that causes the cell to undergo lysis, and it helps kill the cell. Now, again, not all antibiotics kill bacteria. They may just weaken them to the point where our own immune system then can come in and wipe them out. So if it's weakened the cell wall enough, our immune cells can come and phagocytize those cells and basically eat them. If that peptidoglycan is intact, a lot of times your cells, your immune cells, may not be able to completely phagocytize that cell or break it down. So these antibiotics help weaken it, and then our immune system comes in and does the final killing. So this is the NAG and NAM. You don't need to know all the chemical formula stuff here, but just realize that these repeat themselves, and I'll show you a better picture of peptidoglycan in a minute. But you'll hear that term over and over and over again. Peptidoglycan, it's the cell wall. That's the component of the cell wall. And again, this is going to determine which antibiotic can work on a particular bacteria. So when we look at cell walls, it's basically these repeating rows. And if you look, we repeat over and over, NAG and NAM, NAG and NAM. In fact, you can see it better here. So NAG's in brown, NAM's in pink. And again, it's this cross-linking right here that penicillin interferes with, and it helps break down the cell wall because it interferes with the linking between the different strands, right? So this is kind of like the carbohydrate backbone, and then we have these peptide side chains that hold them together. Well, penicillin comes in with the squares that and basically breaks that apart, right? And that's the way that particular antibiotic works. Now, other antibiotics do other things. They may interfere with transcription or translation. It may interfere with the ribosome. It may interfere with the cell wall in this case. But there's all kinds of different antibiotics, and we'll look into those and how they work later. But for right now, just realize that this is peptidoglycan, and this is what the cell wall is made out of. Now, if we look at gram-positive versus gram-negative, gram-positive is going to have a thick layer of peptidoglycan and teichoic acids, and this regulates cation movement in and out of the cell, and it provides the cell wall antigenic specificity, which means we can pick up on it as an antigen. Our immune system can pick up on it, right? The gram-negative cell wall has a thin layer of peptidoglycan, but it also has an outer membrane, so that now is protected. So if I give you penicillin and you have a gram-negative infection, it's not going to do anything because that outer membrane is going to protect that peptidoglycan from the antibiotic, and it's not going to be able to get to it to break it down, right? So again, we'd have to use a different type of antibiotic here, whereas we could use penicillin-based antibiotics on the gram-positive cell wall. Right? So notice the thin layer peptidoglycan. I'll show you a picture of it here in a minute, and you'll be able to see it. These teichoic acids are going to basically link the peptidoglycan, and they carry a negative charge, and that's going to be important when we're staining. If you remember, like charges repel each other, you're going to do a stain called a negative stain, where it's actually going to, the stain is going to be repelled by the bacteria, and the bacteria will appear as basically a uncolored spot on the slide. The background of the slide will be stained, but the bacteria will appear white, right? So this negative charge is going to be responsible for that. Right, so if we look at our peptidoglycan, and we look at a gram-positive cell wall. Notice this is a fairly thick layer of peptidoglycan. Again, it's cross-linked here. And then we have these teichoic acids that kind of help bind all of this together. Now, these can be picked up by the immune system as well. So our immune cells can look at this, and it's antigenic, which means we're going to produce antibodies towards it. And the immune cells are going to react to it, right? But notice how thick this layer is. There's no outer layer. So gram-positive, this peptidoglycan is just laying bare out there. So penicillin, for instance, can come in easily and break these bonds in the peptidoglycan and break down that cell wall. And again, that's what leads to the bacteria's death. When we look at a gram-negative cell wall, you're going to notice there's an extra layer on the outside. So here's our gram-positive down here. Notice how thick this layer is. The peptidoglycan in a gram-negative is very small, and we only have one layer. But notice we have this extra layer on the outer membrane. So this will protect this peptidoglycan from penicillin, right? And so penicillin can't penetrate. It can't go in and do anything to the peptidoglycan. Therefore, it's useless against these gram-negative bacteria. This one, completely exposed. So penicillin can come right in, break this down, and break the cell wall apart. Okay. So some of these polysaccharides that are on the outside of a cell, on a gram-negative cell wall, can act as an antigen, right? And that allows the immune system to pick up on it, right? Also, they can produce endotoxins, which are what makes you sick. So the O157H7 E. coli will produce this endotoxin, and then that will make us sick, whereas regular E. coli does not. We live in a good symbiotic relationship with E. coli. It's just this one strain that causes that endotoxin to form, and that can make us ill. If we look at the cell wall here of a gram-negative bacteria, notice we have a single layer of peptidoglycan. We have this extra layer here, and then we have this periplasm in the middle. That this is where the toxin is formed, right? So the endotoxin is this little blue guy right here embedded inside of the cell wall, and or the not cell wall, but in this case the outer membrane of the cell wall. But these are the toxin producing areas, and then that's what makes you sick. Right? Now, when we look at gram-positive versus gram-negative, I'm just going to skip over because there's no reason to go through this. You're going to do this in lab, all right? So I'm just going to show you kind of the end result of this. But basically, you heat fix your slide. Then you're going to put a primary stain on. Then you're going to put what's called a mordant, which is the iodine. That's going to lock in that stain. And then you're going to take a counter stain and then stain it again. And if it's gram-positive, it's going to appear purple. If it's gram-negative, it's going to appear pink, right? Now, depending on how good you are and how long you leave these things on, you should get some good results where you have all your cells are purple or all your cells are pink. Now, if you don't do the technique correctly, you leave things on, especially like the alcohol, if you leave it on too long, you'll wash out a lot of the stain and you won't see the gram-positive like you're supposed to, all right? So be very careful with the timing. I'm sure your lab instructor will give you specific instructions about the alcohol. I typically tell my students no longer than 5 seconds, right? If you leave it on longer than that, it just decolorizes everything and you're not going to see what you're supposed to see. So follow the directions, make sure you time everything perfectly in lab, and you'll get these results that you're expecting where you have purple and then the pink for gram negative. So gram positive, purple, gram negative, pink. And that's the easy way to remember it. Positive, purple, PP, right? Keep it right in your brain so you don't get mixed up on those. Now, the gram-positive cell wall, again, has two rings in basal body of flagella. These can produce exotoxins, high susceptibility to penicillin, disrupted by lysozyme. Whereas over here, we give the gram-negative, we have four rings in the basal body of the flagella because it has to go through that extra layer of the cell membrane. And these can produce what's called endotoxins and exotoxins. But there's a low susceptibility to penicillin. It just can't get in there and break apart that peptidoglycan like it can on a gram-positive cell wall. This is just another one you can read through if you want to. It just tells you the difference between gram-positive, gram-negative. Pretty much the same things we just went over. Now, some atypical cell walls. You're going to do another staining technique that's called acid fast. And this is going to look for something called mycolic acid in the cell wall. And these are your mycobacterium. And these are some of the nastier guys that you're going to deal with. So tuberculosis, leprosy, these are going to be caused by mycobacterium. Nocardia is another one that will show acid fast. But these are going to show up and you'll be able to recognize that you have a mycobacterium. So let's say you're unknown that you get in lab. You don't know what they are. You get two bacteria. Well, if you get one that's acid fast positive, then you automatically know which one you have. It's the mycobacterium, all right? And it's M-SMEG is the one you're going to use in lab. All right. So if you get that one in your unknown, you do your acid fast, immediately you'll know whether you have that bacteria or not. Because it's stained. If it's positive, you automatically know that it's a mycobacterium. The only ones that will stain that way. Again, I'm not going to go through the technique with you. You'll do this in lab and you'll be able to figure out if you have an acid fast bacteria or not. Right? So if you look at your acid fast, it's going to appear red. Right? So if you do your acid fast and you don't see red, that's negative. If you do see red, then that's going to be positive. Right? This is a really tricky one because a lot of times some of this dye will be left over and you might get fooled thinking it's positive when it's really not. You have to make sure it's bacteria that is stained because sometimes if you get a little agar or something in there, it'll hold on to that stain and it may show a false positive. So you got to be really careful, look really careful with your microscope. Make sure that you're looking at bacteria cells. Look for that shape, all right? That way you know that it's bacteria and not just some trash they got in there that stained. I've seen that happen a lot. Now, mycoplasmas are the smallest bacteria, and these do not even have a cell wall. And they have a plasma membrane. And then again, archaea or vallus, or if they do have a cell wall, it's going to be made of pseudomerium, right? This does not have the peptidoglycan like bacteria do. Again, they're completely different domain. They separated a long time ago, and these two don't have anything in common, even though they look alike, right? Well, I say nothing in common. They do have in common that they're both prokaryotic cells, but other than that, they don't have anything else in common, right? They were lumped together because no one knew anything about them until recently. And then again, with genetic testing, molecular biology, we were able to figure out, hey, these two guys aren't even closely related to each other. They branched off a long time ago. Now, protoplast and spheroblast, these are cells that are kind of abnormal. They get the cell wall gets damaged. And sometimes they don't have a cell wall or lysis doesn't completely occur. So again, these are damaged bacteria, but they look different than the other bacteria do, right? So once they get into these phases, these are easy for the immune system to knock out. So again, I may give you an antibiotic. It may not kill the cell, but it weakens it to a point where it becomes a protoplast or a spheroblast or an L form, and then our immune system can come in and finish the job and get rid of it. All right, so plasma membrane, very similar to what we've seen in the human cell. So when you have an anatomy and you study the eukaryotic cell, basically you have this phospholipid bilayer, and then you have peripheral proteins on the membrane surface, and then you have the integral proteins that act as channels that allow enzymes and things to move in and out. It allows certain substances to get in the cell, certain substances to get out of the cell. We do have glycoproteins and glycolipids that are attached to the cell, and sometimes these can be antigenic, which means our immune system can pick up on those and read it and say, okay, this is not human. I need to get rid of this. Right? So again, this is part of the plasma membrane, not the cell wall itself. Right. So inside here we have plasma membrane, and then we have our cell wall, peptidoglycan. And again, this is going to help protect the cell and prevent it from lysing. So this is our plasma membrane. Again, just like in humans, it's a lipid bilayer system. Remember, the hydrophobic and hydrophilic ends line up. The hydrophilic is going to be this polar head. It's going to face where water is located. And then the tail is hydrophobic. It's going to turn inward. And then we get this two-layer membrane that we see here. We have the integral proteins that act like pores and channels, just like we saw in the human cell. So the structure of our plasma membrane is the same. It's that fluid mosaic model. The membrane is as viscous as olive oil, and proteins move freely for various functions. We have phospholipids rotating and moving laterally. If you take and you disrupt the cell membrane, it'll come right back together and realign exactly like it was here. So let's say I penetrated this with a pin or something. It would tear open, and then it would just reform right back behind it because of that hydrophobic and hydrophilic end of these little phospholipids. So it's always going to orient itself this way, but think of it as a fluid, right? It's constantly moving around, right? It's not stagnant. Right? So selective permeability, just like we saw with the human cell, it allows some molecules in and not others. Right? It allows some molecules out and not others. We have enzymes for ATP production, just like we saw in the human cell. But some of these have photosynthetic pigments, and we'll look at those cyanobacteria later. We'll talk more about photosynthesis and bacteria. But these have chromatophores that are going to act as pigments that capture light. And again, they can use that light for energy. And there you can see the chromatophores. Again, these would be involved in photosynthesis in some bacteria, not all. Right? Only some can carry out photosynthesis. So damage to the membrane by alcohols, quat or quaternary ammonium detergents, and polymyxin antibiotics, this is going to cause the cell to basically leak. And it's going to open up a hole. And you can see down here, this cell, all of its contents inside are leaking out. And eventually that cell will die. So a lot of times when we use antibacterial soaps, antibacterial sprays, different things like that, it's going to contain these alcohols, these quaternary ammonium detergents, polymyxin antibiotic, and it's going to break that cell apart so that it bursts open. So a lot of the antimicrobial soaps and things like that will work this way. All right, so just like in the human cell, we have passive processes like diffusion. So things are going to automatically move from high to low concentration. And then we have active processes that require ATP to move things against their concentration gradient. So hopefully most of this stuff is kind of reviewed to you because it's the same as we studied in anatomy, at least the very first part. It may have been a while since you had it, but hopefully you still remember some of this stuff. So simple diffusion is just simply moving from an area of high concentration to an area of low concentration, and this will continue until the molecules reach equilibrium. right? So as long as we got more stuff on one side of a membrane than the other, it's going to tend to move down that concentration gradient and move in. Now, this is assuming that this can make it through this lipid bilayer, right? If this is a lipid soluble molecule, it'll just move right through. If it's not, if it's water soluble, it's going to have to utilize one of those channel proteins to get in. But it will still move down its concentration gradient. It's just it has to have a channel open up so that it can move through. There's an old term, like dissolves like. So if this is a lipid, it'll move through this lipid bilayer very easily, right? If you remember back in anatomy, we studied the hormones. We had the amino acid based hormones that were water soluble. They had to use the second messenger system, whereas the steroid could just run right through the plasma membrane, the nuclear membrane, and go right into that DNA and switch it on, switch it off. Same thing here. If these are lipid soluble, they'll just move right through. If not, they're going to require a channel to get through. So facilitated diffusion, this is where we have a transporter protein in the membrane. So things that can't get through the membrane that are water soluble or too large, this will open up and allow them to pass through. Right? Just think of it like a gate, and it opens, and then now those big, large molecules can get in, or those insoluble molecules can get in. Right, so here you can see a non-specific transporter. Basically, it just opens up and anything can go through, right? So we call this facilitated diffusion because it's helping. This membrane channel is going to open up and help these molecules get in. If it's a specific transporter, it's going to have a receptor site for a particular substance. So in this case, let's say this is glucose. It's going to fit right here, and then this channel is going to open up and release it to the inside. But it's the only molecule that can get in. It has to have that particular shape in order to get in. So again, we call these specific, whereas these are non-specific, where they can just allow anything in. Now, osmosis is simply the movement of water across a membrane. And again, water is going to move down its concentration gradient, just like anything else. So if there's more water on one side than the other, the water is going to move from the area of high concentration to an area of low concentration. Now, a lot of times we'll have something called aquaporins, or these water channels. We saw this in the human cell as well, but these are channels specifically for water that allows water to move into the cell. Remember, it's hard for water to move through that lipid bilayer. It can because the molecule is so small, but it helps to have an opening like this aquaporin to let it really come in and quickly. So again, some of the water molecules can squeeze their way through, even though it's water and lipid. This molecule is so small, it can just kind of squeeze in there and get in, but it's a slow process. If we want it to be fast, we have these aquaporins that open up and allow that to move right in. Osmotic pressure, we studied this in anatomy. We talked about, hypertonic, hypotonic, isotonic. It's no different for bacteria. They have to adjust to live in certain environments. And certain bacteria can live in salty environments, for instance, the halophiles. Others cannot, right? But it's this osmotic pressure is going to build inside the cell. If it builds too much, the cell can burst open, right? Or if it works the other way, the cell can shrivel up and shrink if water is moving out of the cell too quickly, right? So it depends on what type of environment the cell is in as to what will happen with the water content in that cell. Okay? This is just a simple experiment to show osmosis. Basically, you put a bunch of stuff inside this little sac, the cellophane sac, and then water is going to want to rush in to equalize. And notice over here, when it swells and there's the same amount of water on the outside as the inside, then that's going to reach equilibrium and stop at that point. So again, back to the same terms we used in anatomy. Isotonic, that's where we have an equal amount of solute inside the cell as outside. Hypotonic, the concentration is lower on the outside of the cell and water is going to move in. Hypertonic, you have more solute on the outside of the cell and water is going to want to move out. Again, hopefully this stuff is all reviewed for you. Okay. So isotonic is what most cells prefer to be in because it's a balance and they don't have to worry about water going in or going out. Hypotonic, again, the cell has to worry about water rushing in and causing it to swell and burst. Over here, we have to worry about shrinking and water leaving the cell. Right? So hypertonic, you're going to have a lot of stuff outside the cell and water is going to want to leave and cause that cell to shrivel. Now, with bacteria, since they have a cell wall, it makes it where they can shrivel up like this and survive because the cell wall kind of protects them. Same thing over here, that cell wall protects them to some degree from swelling too much and bursting. That rigid cell wall kind of holds it in place. But if it gets enough pressure in there, it will burst open. So active transport, again, is going to require ATP, and it goes against the gradient. And then group translocation requires a transporter protein and PEP, and it's going to alter the substance as it crosses the membrane. So as it moves through the membrane, it's going to actually change, and molecular level, it's going to change and become a different compound. Right, the cytoplasm, this is the substance inside the plasma membrane. It's about 80% water. You have some proteins, some carbohydrates, lipids, and ions. And then we have a cytoskeleton, just like we do in other cells, that are going to kind of give the cell its shape and its rigidity. And then the nucleoid, remember, bacterial chromosomes are circular, and it's a circular thread of DNA, and this contains the cell's genetic information. So everything the cell needs to survive is located on that bacterial chromosome. But remember, they have this extra bit of chromosome, and that's called the plasmid. And this is going to have non-crucial genes, things like antibiotic resistance, production of toxins. These can usually be shut on and shut off. But this is like a little extra piece of DNA that it can play around with. And again, it can have non-crucial genes they don't have to have to survive, but can increase their survivability. So if I have antibiotic resistance and I am in the presence of an antibiotic, then that's going to benefit me. It's going to help me to survive. So these plasmids, again, have extra DNA that can be switched on and switched off when the cell needs it. Now, if we look at the ribosome, and don't get confused with these numbers, you might see 30 and 50 and say that should equal 80. That's not the case. It equals 70. All right. This is called sedimentation rate. That's how fast it settles out of solution. And this one settles out at 30, this one at 50. But when they're together, they settle out at 70. And that's going to be important that you remember we have 70 S ribosomes in bacteria. All right. So this is basically a unit based on the sedimentation rate in a centrifuge. It's not about size, so you can't just add these two together. This as a whole will settle out at 70 S, right? And that's a Svedberg unit. It's just a unit to show you how fast things settle out when it's put in a centrifuge, right? But do remember that this 70S ribosome is different than the eukaryotic ribosome. When we get to antibiotics later, we'll talk about some that act upon the 70S ribosome, but again, leave the 80S ribosome alone in your eukaryotic cells so that you take the antibiotic and it only kills bacteria, and you have few side effects because it doesn't do anything to your cell because you have an 80S ribosome. Some inclusions, again, these are not in all bacteria, but some bacteria have these specialized inclusions. Some of these have metachromatic granules. Basically, they give it color. We may have polysaccharide granules that are energy reserves, lipids, sulfur that are energy reserves, carboxysomes, which is an enzyme for CO2 fixation during photosynthesis. If it's a photosynthetic bacteria, you can have gas vacuoles to help them float, maintain buoyancy. These are bacteria that live in the ocean typically. And then magnetosomes are iron oxide inclusions, and this helps to destroy a toxic substance, peroxide, right? So it helps to break that down. You're also going to see that some of these bacteria have what's called catalase, and catalase is an enzyme that will break down peroxide, and you're going to take hydrogen peroxide and put it on your cultures to see if it's catalase positive or not. If it bubbles, it's positive. If it doesn't bubble, it's not positive. And that's going to be one way to distinguish what bacteria you may have. So again, you can see those here. And then endospores, you are going to test for these. You will do a stain. They will appear green after you stain it. And Bacillus and Clostridium are well-known sporulators, which means they form endospores. And the endospore is basically just some DNA, and it locks itself up, and basically it's just a little bit of DNA with the capsule around it. And an endospore can last for years. In fact, they think when they went in King Tut's tomb, people started dying. They think there may have been bacteria in there that were in the endospore state that had been that way for thousands of years and then became vegetative again and started to make people sick. So these can last for a long time, and they are resistant to desiccation or drying out. They're resistant to heat, chemicals, radiation. This is a big one when you're canning food to worry about, Clostridium botulinum that causes botulism. If you do not get that canned food up to an extremely high temperature, you have to put it in a pressure cooker and get it well above the boiling point to kill these endospores. If not, if you get an endospore in there, it will germinate, and then it starts making a toxin that is deadly. And you eat the food, and that's it for you, right? So Clostridium botulinum, if you ever get a can and it appears like it's bubbled up or it's swollen, definitely do not eat it. I don't even recommend buying cans that are bent, right? Because they could have tiny little cracks in the can and allow some of these to get in there, and it can make you very sick or kill you. I know some of these thrift stores and things sell, food like canned food that's been damaged, and they sell it for, a quarter or something like that. But I just don't trust it, because if it got a little crack in it and some of these bacteria get in there, it can be bad news for you. So here's the endospore again. When you do your stain, they will appear green. They'll either be inside of a cell like this, or they'll actually be ejected already. It depends on how old the culture is. So you'll have one that's 36 hours, one that's 72 hours, and you'll compare the two, and you'll be able to see where the endospores are located. So the longer it sits, the more likely the endospore will already be ejected out of the cell. If it's a young culture, then the endospore may be inside of the cell. Right. So, and when it forms an endospore, again, the plasma membrane is going to surround the DNA, and then the spore septum surrounds the isolated portion, and then a peptidoglycan layer forms, and then eventually it will be ejected from the cell. And again, this can sit around for a long time, and I'm talking thousands of years. In fact, they've even found endospores that were millions of years old, and were able to get those to become vegetative again and come back and form a living bacteria. So these can last a long time and last in harsh conditions and come back. But you'll do this stain more towards the end of the semester. Well, not really. You'll do it towards midterm and right before you do your unknown. This is a eukaryotic cell. I'm going to leave it to you to go back and review this. This is the same thing you did in anatomy. No reason for me to go over all of this stuff with you. Right. Now, when we look at flagella and cilia on bacteria, flagella are much longer. Cilia are real short projections, but there's a lot of them. Right. Again, they're both used for movement, but one is going to be a lot longer, the flagella, and then the cilia is going to be a lot shorter. All right. So with these, I'm just going to skip over. You should know all of this stuff already. With the organelles, if not, go back and review them. I'm not going to test you heavily on the eukaryotic cell because this is micro, but mainly know the prokaryotic cell. What's the parts of it? What do they do? That type of thing, right? So when we look at eukaryotic plasma membranes, they're similar to prokaryotic cell membranes. Again, it's a phospholipid bilayer. We have those peripheral proteins and integral proteins. Differences, we have sterols and carbohydrates for attachment and cell recognition. So there are some differences between them, and I think there's a chart in here. This is one I definitely want you to know, that eukaryotic are going to consist of 80S ribosomes, and then the 70S ribosome is going to be found in bacteria, right? Now, the endosymbiont theory is that the chloroplast and mitochondria are thought to have been bacteria, free living bacteria at one point, and they still retain that 70S ribosome. That's one reason we think that they were bacteria, because they have the same exact type of ribosome as a bacteria cell. So think of a chloroplast and a mitochondria as kind of a cell within a cell. So in our cells, we have these things. Well, plants have chloroplasts, but we have mitochondria. They do their own thing. They make their own energy. They have their own DNA. They have their own ribosomes. So again, they're almost like a cell within a cell. Nucleus, again, we've went over all of that stuff in anatomy. I just expect you to review these, all right? So I'm not going to go into detail with all of this stuff because you've already done this. All right. So when we look at eukaryotic cells, it's thought that they originated as prokaryotic cells, and then they first evolved around 2.5 billion years ago to become the eukaryotic cells that we see today. So life arose about 3.5 to 4 billion years ago, as best we can tell. And then the first eukaryotes didn't appear until 2.5 billion years ago. So again, the prokaryotic cells came first, and then the eukaryotic cells developed later, right? And so the endosymbiont theory, the larger bacteria cells engulf smaller ones, and this developed the first eukaryotes. And ingested photosynthetic bacteria became the chloroplast, aerobic bacteria became the mitochondria, right? So again, it's thought that these were once free living and became part of the cell, and then over time they evolved to become part of the cell. So again, it's thought the mitochondria and the chloroplast were free living bacteria and then became part of a cell, and then that became the eukaryotic cells that we see today. Right? Now, if you look, you can see archaea split off way back here from bacteria. So again, they're not related, even though they look alike and they're both prokaryotic. This lineage went down another pathway, and they basically stayed the same, and they didn't really change all that much. But this bacteria eventually became eukaryotic cells that we see today. Right, I did get through. I didn't think I'd make it. So we'll still keep your test for Sunday. Make sure that you get all of those handouts, and I'll post all of the recordings here shortly. I think everyone's been posted except for this one. But this is the sheet I was talking about in chapter 4. It gives you all the different shapes. It gives you the arrangements, and then it gives you the arrangement of the flagella on the cells. All right, so you can find that one there. It's right here. So go under handouts, chapter 4, and if you'll go here and go to handouts, again, you'll find that sheet to help you study. And then if you want to do this, go to chapter 1 handout. You have to do the timeline. Where did I put the other one? Hold on. Let's see. There's your extra credit for the bacteria cell model, right? Also, there's a Gram stain handout. It basically is going to take you through the Gram stain technique and show you how to do it. Follow your lab instructor's guidance on this, because they may tell you to do alcohol for only 5 seconds instead of 5 to 10. They'll give you their own. I just wanted you to kind of get an idea of how it's done, so you can look at this ahead of time and kind of see what you're going to be doing in lab. All right. All right. I think that's all I got for you today. I didn't think I was going to get done, so I didn't prepare a quiz. So you don't have anything to worry about. So you're going to get out a little bit early today. I'll hang out for a few minutes. If you have any questions, you can either hit me up on the chat or just ask me either way. So anyway, if you don't have anything, y'all are good to go. And again, I'll stick around for another minute or two and make sure nobody has any questions about anything. I got you, Loretta. You're on here. 3 and 4, yes, that's correct. Look at the 19th, I believe. Let me go to the calendar. Should be sharing this with you anyway. There's 8 more assignments, 8, 9, 10. There's 11 things that'll be due on that Sunday, the 19th. Don't forget when it says 8 more down here, there's more stuff in here. So when you click on it, it'll open those up. So if you click here, you'll see all of it down at the bottom. Hopefully you've done some of this stuff already and you won't have to catch up on it, but everything is here. Yes, this test is Sunday on Respondus. It's open from midnight to midnight. So you have 24-hour period to basically get it done. So if you work weird hours, it'll be open in the middle of the night, right? Or if you want to do it in the day, you can do it during the day. But yes, you'll need the lockdown browser. So make sure you test that with the quiz, the lockdown browser quiz. Make sure everything's working properly. I'll try to check my email Sunday in case people run into problems, but I can't be here all day looking at it. So. No, once you start, you need to finish it. It'll be timed, so it will run out on you. Do that to prevent cheating. I can't tell you right now how many questions are on there. I think I haven't loaded it yet, but I want to say it is around 60, somewhere around there. But don't quote me on that. It should be close to that though. Multiple choice and matching. Only multiple choice, but there are some matching questions too. I'll try to post a study guide, give you an idea of what to look for. I'll try to get that up tonight. They got me teaching a Friday class now, so I've been really busy. It's just hard to keep up with everything, but I'll try to get that posted tonight when I get home. All right, anybody got anything else? I'm gonna close out. You're welcome.